Sophia. Thank you very much for taking the time to speak to me today. And I've prepared a few questions. You are doing something very exciting within the area of cancer immunotherapy. And I will be very pleased to kind of like hear more about your research and what the group is involved in. As you know, as a starting question, would you be able to give me a bit of more information as to what the group is involved in, what your research focuses on at King's College London?
Of course. The key areas of research in our laboratory include the study of cancer patient-derived B cells and antibodies, and Fc receptor expressing immune effector cells in the tumor microenvironment and in the circulation of patients. Alongside engineering antibodies of any specificity, class or isotype, including Fc engineered antibodies, and also we have a program on antibody drug conjugates emerging in our lab.
That's great. How many people are working in the lab and how many people are working in your group? How big is the group?
We range between 12 and 15 people, depending on the season, and people come and go, finish their PhDs, new people are coming, new postdocs arriving. So we're quite a dynamic group, as I think all academic groups are, very much translationally focused and very much looking to see what we can do to develop antibodies that can go from concept to the clinic and beyond.
That sounds really exciting. As you mentioned, one of the key areas of your focus is cancer immunotherapy, that the group's focus. Why have you decided to study this area specifically? What makes it so exciting?
I always believe that the immune system is a protective mechanism for many diseases, and I could not see how this could not be relevant in the context of cancer. I've been studying antibodies and B cells since I was a PhD student, and I've always had a passion for trying to understand the interface between antibodies and immune cells, especially immune effector cells and cells that actually express them, such as B cells, and how they activate immunity and how they engage with cancer. So my interest started looking at more basic mechanisms of antibody and effector cell mechanisms, and then move on to consider how could this be applied in the context of cancer. I think our immune system is a very powerful tool. I think a lot of evidence now tells us that these mechanisms that a tumors trigger can suppress that immune response. We need to understand why, and then what can we do to wake up those responses and divert them to target cancer.
And are there any other areas as well that the group is looking at, or is it predominantly cancer immunotherapy that is the main focus of the group?
So by and large, we always come from the point from immunology perspective rather than from the cancer perspective. So we are very much interested in trying to understand how immune cells work and how different antibodies and different antibody isotypes engage with these immune mechanisms. And then we look how that applies to cancer. We also have a program in our lab very much interested in immune markers and biomarkers, and also cancer biomarkers and how these crosstalk with immunity as well. So again, like I said, it always comes from the immunology perspective, but we do reach out to cancer a lot. And we're very privileged to have collaborations and bonds with a lot of clinical academics and clinical teams. And we also are linking with a lot of very talented and forward-thinking researchers around the world. And we think that group together, that nexus between us and others, that link really brings translational research forward. We also are very much out looking at innovation and thinking about linking with commercial partners. And we have a few commercial links, which I think is really crucial to be able to move forward innovative therapies, to assist them for their innovative platforms and therapies, and they can assist us and they can inform us on how to move forward towards the clinic and beyond.
Yeah, certainly. That sounds very exciting. I'm sure that like with any other area, there are a lot of challenges that the group is facing and you are trying to overcome. So what would be the biggest ones that you are trying to overcome right now? Are there any steps taken that the group is trying to adopt in order to overcome some of these challenges?
I think, but generally scientists are very resourceful. and very resilient. And I think talking about this at this time, we cannot help but discuss COVID and the crisis that we're facing. I think science has really stepped up in this pandemic, and a lot of people have pulled together to help and to move the science forward, to develop therapeutics and vaccinations that can make a difference for patients. So the challenges have been, of course, access to the laboratories and the issues around the pandemic. That has been a very big challenge for us in the last year, on top of all the other challenges that normally science faces. But I think a lot of people have taken a moment, they have consolidated, and they have come up with new ideas. They have been very productive during the last year. And I think that's actually going to help going forward, spur research on going forward. We can turn the negative into a positive, I think, in many ways.
Exactly.
I think another thing that's come out of this situation and the pandemic is that the public has been now more aware of who we are and what we're trying to accomplish. The negative things about the last year has been that from our perspective in cancer therapy, research for cancer has taken a slight step back, and we think we need to ramp up the efforts moving forward, because we still need to help patients, and cancer is not going to go away without us working super hard, and we're committed to do that. I think everybody in the lab is committed to do that.
Yeah, exactly. How interesting that, the focus can change from one day to another. We have this pandemic going on, and I think every research group out there needed to adjust their plans and shift their focus towards looking at the pandemic as well. And obviously this comes with a lot of opportunities, but as well a lot of challenges that need to overcome.
Absolutely.
Yeah. Well, Sophia, you will be giving a talk at our event in April, which we are very excited about. It's going to be on Ig class antibodies for cancer immunotherapy. What are you planning to touch upon in your presentation?
Thank you for bringing this subject up. It's one very close to my heart and a very big focus in our group. We come from the perspective of the immunologist. And in fact, I have worked many years on Ig immunology, and mostly in relation to allergy. But it became very clear to us that there's a lot more to the immune response and to antibody therapeutics than the classically used IgG. As we all know, monoclonal antibodies approved for the treatment of cancer are largely of the IgG class, and those that are expected to have FC mediated functions are commonly of the IgG1 class, the most common antibody class in the human circulation. So we hypothesized that exchanging the Fc regions of an anti-cancer IgG antibody with Fc regions of the IgE class, an antibody class known to exert immunological effect in tissues through very high affinity for cognate Fc receptors in tissue immune cells, such as macrophages and mast cells, might help recruit and activate the cells against tumors. Interestingly, most people consider IgE a pathogenic contributor in allergy. However, IgE is also a protective antibody in our tissues from parasitic infections. And in fact, it has these types of effector functions we are after to trigger in cancer in the context of parasitic infection clearance. So therefore, we wanted to know if it's possible that the same mechanism can be triggered against cancer. At the time we were doing the work, we were not sure whether we will activate an allergic response or antiparasitic response. And interestingly, we actually found that IgE can kill tumors by harnessing known immunological mechanisms it naturally employs in parasitic infection clearance. And we found that IgE potentiated, interestingly, monocytes and macrophages functions and recruitment and re-education from alternatively activated phenotypes to a more classically activated phenotype, one that resembles the effector functions we see against parasites. So if we have worked from the concept until to the clinic, we have designed the first-in-class Ig therapeutic candidate, which is now undergoing and almost completing clinical testing in oncology. This to us will lead the way for new opportunities to extend the current IgG-only pipeline for the treatment of cancer patients. We'll learn a lot from this, and we have taken, because we've taken the concept from early development to the clinic, we were able to feel the pipeline and try to understand how we can engineer, how we can develop an antibody, how we can make it safe, and we can produce it to be safe to go into patients. And we have now had a chance to give it to patients. We have experience in how this antibody behaves in patients when it's given systemically.
Yeah, that sounds very interesting. Thank you very much for sharing this with us. We very much look forward to your presentation.
Thank you very much. I'm always very interested to hear also the feedback from the audience and to have a very interesting discussion.
That's great. And now I would like to conclude this interview with a fun element. It's a turn of quick fire questions. So I prepared a few questions, and the first thing that comes into your mind, please just feel free to say it. So, are you ready?
Yes.
Where will you go on holiday once the pandemic is over?
I will go immediately to see my mother in Athens and to spend some time with my family, my sister and my brother's families. I miss them tremendously. Yeah. It's been a year, and it is very painful being away from them.
Yeah, I feel the same. My family is in Eastern Europe as well, and I very much miss them. What's your favorite day of the week?
I think it's Friday. So today is Friday. It's just when you conclude all the work and you think you've accomplished something for the day.
Yeah. What was the last film that you watched?
I watched Pretty Woman recently.
What is your favorite cuisine?
Wow, that's a difficult one. Oh, wow, I like so many. I would immediately say Italian.
Italian. Do you prefer summer or winter holidays?
Definitely summer.
Yeah, that's my preference as well. Great. Sophia, thank you very much for taking the time once again to sit down with me today to talk about your work. And I wish you and the group good luck for your future plans. And we are very excited about your upcoming presentation at the event.
Well, thank you very much for inviting me. Thank you for inviting me to the conference, and I'm very much looking forward to participating and interacting with participants and lovely speakers you have invited.
Great. Thank you very much.